Today we were talking about cross-linking cell surface glycans with lectin protein. And my name is Yang. This is Lam. The glycans are a combination of sugars of cell surface in all eukaryotic cells or cell extracellular proteins to bind with other particles such as this HIV virus or other protein, enzyme, or bacteria or other cells. This nice structure are nice sugar binding blocks in humans, and all glycans in human cells are actually combination of a random or particular sugars from none of them to form the structure. And also we're gonna using mannose, gal NAC, and the other, I think, galactose as an inhibitor in the later of this lab to test for the inhibitor rate of the thing. Lectins can actually usually can be found in plant such as the beans that we were using last week. We grinded the bean to obtain lectins by dilute the grinding little particles into a buffer. And lectins is actually multi valent proteins that will bind with glycans. And remember, glycans are really good at binding. We will find our glycans from blood cell from certain animals, and we had five choices. We had a cow blood, pig blood, sheep, horse blood, and rabbit blood. And in my group, we were actually using the cow blood. This is an example of a convenient assay for glycan hemagglutination. When we see the red dot right here, means that all the red blood cells are actually at the bottom of the well, and there's no protein to bind with, so it won't be like this. The hemagglutination that the blood cell actually is binding with the protein and stay in on the cell wall, on the well wall, and that's gonna be standard that we're gonna use in the future of our laboratory. The inhibitor test we're gonna use is that we're gonna use one of the sugar from monoscartride. Scartride. The reason that the inhibitor will work because the inhibitor, for example, we're gonna use mono, and perhaps there's a part of the glycan from the blood we were using has a similar structure of mana, so the lectin will likely to bind with the mana before binding with the surface of the blood cell. So basically, the single sugar particle will be a decoy for to occupy the lectin. The lectin eventually will miss the target. And to determine the moment of truth that which inhibitor is actually working better for the lectin is really similar to the experiment we were doing the week before last week, which plot the percentage of agglutination versus the log of molarity or the percentage of the solution of inhibitor inside the solution, and by determining the IC50, the point of IC50, or to determine that which inhibitor is actually more efficient. And this is our first part of the experiment. I put this way so I can enlarge the photo so people can actually see a little bit better. Our A is lactose, and B, the number 2 column is mug, and the number 3 is kidney, and number 4 is A, and number 5 is fava, and number 6 lima. And see, for some reason, 1, 2, 4, 5, 6 has no hemagglutination, so we decide to use a 3, which is kidney, because we can see the number 1 is actually perfectly glutination. The second one is almost, but not quite. And because we're gonna use 10 times of the concentration, so we decide to use the 3D to do our tighter dilution. For this right here, this experience, actually this is a 4 times dilution, which means that it's fourfold that dilution. The first one is actually 1 to 1, and the second one is 1 to 4. This is a 1 to 16 and 1 to 64. That's why when we're gonna do the next step, the tighter dilution, we're gonna use the 1 to 6.4 as the solution percentage, like the volume wise. And this is our 10 times. You can see this is regular right here, and the column above it is actually the heated 10 times. And you can see we start with 1 to 6.4 as a ratio of the solution, and each well from A to H is actually twofold. So basically, it means that this is 1 to 6.4, and this one is 1 to 12.8, and so on and so forth. And you can see clearly that a heated has actually less likely to form a hemagglutination, because when you heated the protein, you actually denature the protein, so the lectin and the glycan will not be function as good as it's supposed to be, because the heated energy is actually overpower the hydrogen bond to actually force them to denature themselves. And that's the result right here, that without heated, the regular tighter dilution actually work much better than the heated 10 times tighter dilution. You can see that the inhibitor hemagglutination versus a log of dilution is totally messed up, because we're supposed to the other way around. And through this graph we're looking at, it actually looks like that is now only that is now working as inhibitor, but it's actually more help the lectin to bind with the bind with the glycan. That's why at the beginning see the most molarity of the inhibitor actually has a better hemagglutination, which is really weird. We consulted with some of other groups' data, and actually a lot of group has the same result as ours. It's really tricky, because I do not know how to explain this. It could happen because different reason that are beyond my knowledge, but you can see that the there's no efficiency of the inhibitor. And the blue color is actually the galactose, and the orange color is the acetyl glucosamine, and the gray color is mannose. And and uh, and that will be all. Thank you, everybody, and we're now open for a question.